Welcome, everyone. Thank you for joining us today, and welcome to the first session of the Clinical Oncology track of our Thermo Fisher APJ Genetic Solutions Tour 2020. My name is Bansi Sangvi, and I will be your moderator for this session. In this session, we will hear about our latest next-generation sequencer, the Ion Torrent Genexus System, and its application in clinical oncology research. First, we will hear from Dr. Jon Sherlock, Director of Global Product Management Oncology, Clinical Sequencing Division from Thermo Fisher Scientific. Jon is based in California, where he runs the Oncology Product Management Group in the Clinical Sequencing Division, developing and managing diagnostic and research products, including our first IVD-labeled NGS kit for solid tumor testing, as well as assays for immuno-oncology, cell-free applications, heme-oncology, and pediatric cancer testing. For a complete biography on Dr. Sherlock, please visit the tab at the top of your screen. Today, he will talk to us about how the Ion Torrent Genexus System will simplify your molecular profiling journey by addressing key challenges in next-generation sequencing implementation, like hands-on time, time to result, user expertise, and affordability. We encourage you to submit your questions in the Q&A box, and Jon will address them at the end of his presentation. Please join me in welcoming Jon Sherlock.
Thank you very much for that kind introduction. I'm going to talk about how to simplify your journey of molecular profiling with single-day next-generation sequencing. As I'm sure you're aware, next-generation sequencing has the promise to revolutionize precision oncology clinical research. With the abundance of information that we have available to us and the increasing amount of exploration and research that's revealing additional genes, be it at the DNA level, at the fusion level, tumor mutation burden, sequential testing for one marker after another is pretty much impossible in this day and age. So next generation sequencing, bringing the ability to bring all this information to bear in one single test, is extremely compelling. However, next generation sequencing, or NGS, isn't available and utilizable immediately for everybody today. Systems can be too slow, requiring days, often weeks, to get a result. The process can be extremely complex, often requiring different machines, multiple touch points, and a high level of expertise to actually run the NGS system. Also, it can be very costly to train staff, to hire staff, and having to batch samples together to reach a sensible critical mass can be difficult and costly. And also, the utility of NGS has been restrained by most platforms requiring quite a large amount of material, quite a large amount of tissue to actually get a result, which can be extremely limiting. I'd like to present to you today a real new dawn in precision oncology with the release of the Ion Torrent Genexus integrated sequencer. The Ion Torrent Genexus system is the latest generation of NGS platforms from Thermo Fisher Scientific. As we go through the rest of the presentation, I'll show you how it's extremely fast. Within a single day, you can go from sample to report. How it's essentially hands-free. There are 2 touch points and only 10 minutes of hands-on time in the entire process. Also, how it can be cost-saving with the flexibility of running a very small number of samples, even a single sample, in a split lane system on the chip, through to running the complete chip. There's a high flexibility. And also the success with tissue saving. How as little as 10 nanograms of input nucleic acid will give reliable results almost every single time. So now with the new Ion Torrent Genexus system, you're able to get results from sample to answer of NGS around the same time as immunohistochemistry. This is really quite revolutionary. With the limited amount of hands-on time, it's very scalable, it's fully automated, very easy to train anybody to operate the system. As I've said, the chips are split into lanes, so this reduces the need for batching, and you can deliver results however many samples you have to process faster than ever before. Although the system is currently sold for research use only, we have built it under a registered facility with the FDA, and it's ISO 13485 certified, which gives us a lot of possibilities for future development. So let's talk about some of the assays that we have available now and those that are coming on the Genexus platform. We have developed it for consideration for both FFPE blocks, for blood, for other tissues, and also cell-free plasma material. We launched the platform at the very end of 2019, and at that point, supporting any custom Ion AmpliSeq HD panel. That's the AmpliSeq HD is our latest chemistry, and also the older AmpliSeq chemistry as well for any custom platform. Also, we have the Ion AmpliSeq on-demand panels. As we moved into the beginning of this year, we have launched a new Oncomine assay called the Oncomine Precision Assay, or OPA. We'll talk more about that in the coming slides. It's a 50-gene marker for use on both FFPE and liquid biopsy samples. We've also released the Oncomine Comprehensive version 3 assay on Genexus. This is a 153 larger gene panel of DNA and RNA fusion detection. And also the TCR beta long read assay, T-cell receptors for translational research in immune oncology. As we move into the second half of this year, we will soon launch the Oncomine myeloid assay and the Oncomine BRCA assay onto the Genexus, with many more assays to come in 2021 and beyond. So a little more about the Oncomine Precision Assay. It is a pan-cancer content of 50 key genes covering all the clinically relevant mutations, the copy number variants, many different fusion variants as well, tumor suppressor drivers, and resistant variants. The assay is designed with a single workflow and SOP to use either FFPE tissue or plasma samples. So with the same lab workflow, you can analyze cell-free material or a tumor biopsy. The approach is to use our AmpliSeq HD chemistry in this assay, which has molecular tagging. This allows an extremely high sensitivity of detecting very, very low levels of variance, which allows the assay to be used for the liquid biopsy application. We've also developed new RNA fusion detection technology we've termed Fusion Sync. More about this in the later slides. So the one assay kit can be used for different applications. If you're interested only in a DNA mutation spectrum for FFPE, you could run 32 samples across the whole chip. Likewise, if you were only interested in the RNA fusion content, you could run 32 RNA samples on the one chip. If you're interested, as most people we believe will be, in looking at both the DNA and the RNA, you can run up to 16 samples across the Genexus chip. That's 4 samples on each of the lanes, or just 4 samples on one lane. It has that flexibility, or even 1 sample. If you're interested in cell-free total nucleic acid, you can run up to 4 liquid biopsy samples on the Genexus platform in one go. The Fusion Sync technology, we believe, brings the most valuable approach to detecting fusion drivers in oncology samples. There are 2 key parameters to consider when you're selecting the technology to use. One is the performance of the fusion detection with very low input of material, together with heterogeneity and also low level of transcripts within the sample. So it needs to be very sensitive. The second is also how comprehensive the approach is, how many different fusions and different fusions from the same driver can be detected. Many approaches emphasize number 2 here, rather than considering both 1 and 2. With Fusion Sync, we address both of these very important parameters. So what is fusion sync? It's a combination of three different approaches. One is targeting known fusions. This is where we have the driver gene and we have the partner gene. And we've trawled through the literature and have many, many hundreds in a regularly updated and curated database. We design specifically across this fusion breakpoint. And so there we would detect the known driver gene, the known partner, and at the known breakpoint. Within the design also, we have designs where we don't necessarily need the exact breakpoint, but we do know the driver gene and we know the partner gene still. We would just have them spanning a larger region and pick up fusions with a previously unknown breakpoint. Both these methods are incredibly sensitive and robust and enable you, of course, to identify the exact fusion transcript that you're detecting, that you're sequencing all the way across. We also have this new approach of exon tiling imbalance. This is where we design amplicons across the length of the messenger RNA transcript. And by looking at relative read amounts, we can detect an imbalance. So here we know only the driver gene. We don't need any additional knowledge, and we'll pick up a fusion event. So the benefits of the targeted approach, as I've said, is that of sensitivity. Here's a couple of studies that we highlight which show how our approach can pick up really low levels of expression. The three different colors here, and this is from a poster from Dr. Song from Cornell at the recent AMP conference. This looks at a dilution series of a known control with the fusion mix contained within it. And as you go through the dilution series, different technologies drop off at different times. Here we're represented with the yellow, with the only approach which actually picked up this fusion at every single dilution. These are different fusions going down the titration series. Other approaches don't have that level of sensitivity, which is really important. Clearly, if you have a low expressed fusion driver, maybe a poor quality sample, a small amount, you need to be confident of picking it up. The exon tiling imbalance approach allows us, as I've said, to detect these novel fusions. So by measuring the expression ratio across the gene, if you have a normal expression, you'd expect a balanced line, and this is what's represented here. So any genes that do not undergo a fusion are expected to have a similar ratio, and we analyze this and we plot it statistically and give a confidence value to show that this is indeed a normal non-fusion event. If there is a break, a translocation, and therefore a fusion transcript that's created, then this expression ratio changes. You can see quite dramatically here how there's a big drop. Importantly, we also map the position of these amplicons according to the kinase domain, because of course you need an active kinase domain to make the driver gene in the fusion pathogenic. So the position of the breakpoint is also very important, and this is revealed in the output of the instrument. So that was a little bit about the Oncomine Precision Assay. Let's move on now to the myeloid assay, which we'll be releasing later this year. So in the past, to detect this growing number of important markers in myeloid oncology, people would have to employ a series of sequential tests, be it Sanger sequencing, PCR, qPCR, or maybe even outsourcing and sending samples out. This, of course, becomes laborious, it becomes very time-consuming and inefficient, losing control of the sample if you're sending it out, and can often take many weeks to get a result. Next generation sequencing is somewhat of a disruptive methodology, as we've said before, allowing you to do all of this analysis in a single run. We have the Oncomine myeloid assay, which we'll be releasing onto the Genexus platform later this year. This has 40 DNA targets and 29 fusion drivers with many hundreds of different partners, and allows the test to be run within a single day. from extraction of blood or aspirate, bone marrow aspirate, all the way through sequencing and out to the report can be completed in a one-day turnaround time. And this is with the simultaneous profiling of the DNA mutations and all of the RNA fusion transcripts into this integrated report at the end. We retain the flexibility of sample throughput as well. And as I've said, we validated the approach on whole blood, lymphocytes, and also bone marrow. Again, it's the same limited hands-on time of 10 minutes for the whole process with 2 touch points, all the way through to the annotated variant report, which lists clinical trials, guidelines, and labels that are appropriate to the mutation spectrum that's been determined by the test. Up to 8 myeloid samples will be able to be run per day. All 8 samples going into a single lane of the Genexus chip. So day 1 would be one lane, day 2 would be the next lane, day 3, and so on. The reagents on the deck of the platform are stable for many weeks. So you retain that flexibility. It's a 2-week guarantee. You can come back day after day and just reload however many samples that you have onto the platform. The purification system, which is launching a little later, will be able to process up to 12 samples at one time with DNA and RNA. This reveals then both an archive plate of nucleic acid and a plate of DNA and RNA that's ready to transfer directly onto the Genexus integrated sequencer platform. This then runs the sequencing process for 4 samples over 17 hours and for 8 samples of DNA and RNA in just over 21 hours. The content of the myeloid assay was selected intentionally to cover a broad spectrum of myeloid disorders. So that's AML, MDS, MPN, CML, as well as CMML and JMML. So we've spent a lot of time and care making sure we selected the right gene content. These are broken up into those genes we cover by hotspots, with those that we have full exonic coverage of the gene. You can see the list here. These are the 29 fusion drivers, as I said, all with many, many different partners with their specific designs. We have some controls and also some expression markers, where by looking at recounts, you can determine overexpression of these relevant genes. All in all, 40 DNA genes. The 29 drivers have more than 600 different fusion isotopes, and we have the 5 expression genes and the controls. Very easy to use, very flexible and robust, and a fully integrated end-to-end solution from specimen all the way through to report. If you compare that to some alternative approaches, we've tried to list here some key factors that we feel are very important to think about. One is the automated specimen to report workflow, where really you don't need any high level of training, you don't need to spend more than a few minutes in the entire process. Another is the speed to result. Obviously, it's critical with myeloid disorders to have information as quickly as possible, the next day, ideally. So having a one-day turnaround time is really significantly important. Next is the content and the detection of both DNA and RNA targets with one straightforward assay simultaneously. Of course, we feel it's important to have the integrated analysis solution all the way through in the software to the report. And finally, it's the coverage for all of the key myeloid targets that you need for a broad spectrum analysis. And the Oncomine myeloid assay for Genexus ticks all of these boxes. So to summarize, we're very excited with the launch of the myeloid assay on the Genexus sequencing system. It will simplify and accelerate myeloid profiling to a single day using next generation sequencing, which will allow laboratories to get rapid results for things like FLT3, ITDs, TP53, CBP alpha, NPM1, KIT, PML, RARA, and all other biomarkers in a single day. Simultaneously interrogate all the relevant DNA mutations and fusion transcripts for research on myeloid malignancies, covering all the major myeloid disorders, AML, MDS, MPN, CML, CMML, and JMML. Even challenging targets like the FLT3 internal tandem duplications are covered. We have dedicated software specifically for picking up these large and unusual events. And the report contains all of the relevant guidelines, labels, and clinical trials. The Oncomine myeloid assay is one of a suite of oncology assays that we have on our older platforms that we're progressively going to bring through to the Genexus platform. They all built on AmperSig technology, with a lot of care and attention on software, the informatics for interpretation, as well as enhanced manufacturing quality controls, making sure that we're producing the highest grade material that goes into all of these assays, with dedicated protocols for use on clinical research samples, and we validate the system and process using the appropriate clinical research samples. We also have dedicated teams to support laboratories implementing these assays. So thank you very much for your attention. I'll finish just with a summary of this really exciting breakthrough in NGS technology, the Genexus system. It's the world's first turnkey automated NGS system, allowing a single day turnaround time. For instance, you can get the full myeloid results within a single day. Automated sample purification, library preparation, sequencing, and analysis. It will vastly increase reproducibility and efficiency of any laboratory whilst reducing personnel costs and training time. It's really flexible with the multi-lane system in the chip. You can run a few to a large number of samples. Reagents are stable on deck. There's negligible wastage. And we've manufactured these in the highest quality systems registered with the FDA and ISO 13485 certified. So as you've heard, we've a growing menu of Oncomine assays we're porting onto this exciting platform. And we're really excited with what the future holds. Once again, thank you very much for your attention. And I believe there'll be the opportunity for questions coming up soon. Thank you.
Thank you so much, Jon, for that wonderful presentation and giving us an overview on the game-changing Genexus system. We will now move into the live Q&A portion of the presentation. As a reminder to the audience, please submit your questions via the Q&A box. Any questions that are not answered today, in the interest of time, will be addressed via email. All right, Jon, we've got several questions coming in. My first question to you, Fusion Sync seems like a very powerful tool. What is the sensitivity or accuracy for the detection of novel fusions such as NTRK fusions using OPA?
Oh, that's a great question. We have had over 99% sensitivity and accuracy when testing the fusions in the samples that we've got through our validation exercises. So it's extremely sensitive.
Thanks for that. There's another question here. What's the maximum output in gigabytes per max reads per run of the new Genexus system?
Right, yeah, that will vary a little from run to run, but you should be sort of 50 to 60 gigabytes on a complete chip. Of course, the chip's divided into 4 lanes, and you can run each lane separately. But if you were to run the complete chip, I believe it's 50 to 60 gigabytes.
Thank you for that. Another question we have here is, what is the recommended minimum tumor content percentage in FFPE samples for using the Oncomine Precision Assay?
We've tested it down to around 10%. That's what we've used as our minimum recommendation. We're generally quite conservative with the metrics that we advise, so I suspect you'd have success going a little lower, but we test down to 10% tumor content.
Great. What is the QC required for the DNA and RNA samples before loading onto the Genexus platform?
You don't need any special treatment. That's one of the great natures of the chemistry and the instrument platform that we use. It's extremely robust. So any standard FFPE sample can be put onto the platform. There's no particular specific special quality controls needed. Just whatever you have as a sample you can put on and expect success.
Okay. We have another question here on the consumables. There's 2 weeks consumable stability seems to be short. Are there any plans to extend that?
Right, yeah. So as you rightly say, the consumables that you put onto the deck of the platform are stable there at the temperatures of a normal laboratory for a period of time. Right now, we're guaranteeing at least 2 weeks. We do expect that to be extended as we continue to test the platforms that are out there and the robustness of the chemistry on the deck. So yes, 2 weeks for now, but we expect that to be extended.
Can you elaborate a little bit more about the assays that are coming forth in 2021 on the platform?
Yes, absolutely. We intend to extend our heme portfolio by bringing additional blood cancer assays onto Genexus in 2021. We also have ambitions of a larger, very comprehensive high gene count, over 500 gene assay that we're expecting to bring in 2021. And some other developments, some MRD kind of applications that we're quite excited by. But yeah, we've still some work to do through this year to see that come to fruition.
Okay, thanks so much, Jon. I think that's been a great presentation. In the interest of time, we will probably end this presentation and move on to our next one. But to the audience, if there are any questions that were unanswered, we will reach out to you via email. So I'd just like to thank Jon again for his presentation and to the audience for all your questions. Thanks, Jon.
Thank you. Thank you very much.
Next up, we have Dr. Michael Hummel, who is the head of the Molecular Diagnostics Lab at the Institute of Pathology in Charité, Berlin, and a long-standing researcher in molecular mechanisms in malignant lymphoma with more than 350 high-ranking publications. For a complete biography on Dr. Hummel, please visit the tab at the top of your screen. Today, he will share with us his experience with the new Ion Torrent Genexus System, as well as presenting evaluation data from a multicentric study on the performance of the sequencer, as well as the new Oncomine Precision Assay. Please join me in welcoming Dr. Hummel.
My name is Michael Hummel. I'm head of the molecular diagnostics at the Charité in the wonderful city of Berlin. And I would like to guide you through this presentation dealing with next generation sequencing and new developments for this application. Now, the disclaimer. So sequencing is not a new technology. It already started 1977 when Maxim and Gilbert reported a new method for sequencing of DNA. But nobody is using Maxim Gilbert because there was a second technology published in the same year from Sanger and Coulson, which was much more successful because it was easier to handle in the lab and to generate a sequencing. So that is also the reason why the number of publications using the Maxim and Gilbert approach, which is time-consuming and which is lab-consuming technology, disappeared almost completely from the scenery. And Sanger sequencing was established. Initially, it was a radioactive labeled system, which was later on replaced by fluorescence labeled dyes. And we were all happy with this technology for many, many years. And it also was the basis for the KRAS testing in colorectal cancer a couple of years ago, when there was an approval of the FDA for anti-EGFR receptor antibodies for colorectal cancer, because there's in many cases an overexpression on the protein level of these molecules. But what was ignored at that time, that the overexpression of the EGF receptor is not sufficient. What we need in addition is information about the mutational status of the RAS molecule. If RAS is mutated, then this molecule is able to initiate signaling into the nuclei and to initiate cell growth. So even with the anti-EGFR antibodies, in the case of mutated KRAS, there was no successful treatment. And it took some time until this testing was established in the guidelines for treating colorectal cancer. And as you can see here from our lab, it's a nice representation of Sanger sequencing with the fluorescence dyes and the detection of KRAS mutation. In this case, the patient will not benefit from the treatment. Continue. Now, a few years later, now we are in the year 2020, there was a lot of development over the last years. And we have a number of already established biomarkers and much more are under evaluation. And even the term biomarker received some addition. So we initially talked about the KRAS mutation. This is the green labeled alteration. But in addition, we have now amplification, we have translocation, we have RNA expression, we have the protein expression, we have some other alterations, I come to this a little bit later, and even non-human DNA. All these categories can be regarded as biomarkers. And if we try to translate this into the cancer types, and the most prominent one at the moment is the non-small cell lung cancer, you can see it's quite colorful. We have a number of different alterations in this disease, and also different categories ranging from immunohistochemistry to translocation, generating fusion genes, and of course mutations. But lung cancer is not alone. We have the gut cancer, and we have many, many other cancers around the body which carry different types of alterations, which are quite, to some extent, cancer-specific, but also there's some degree of overlap across the different cancer types, but with sometimes different functionality. And even breast cancer, you can see the only red-labeled category here, the gene expression. There's a gene expression which is very typical for breast cancer and helps the understanding of this disease much better. So you see, in this scheme a broad variety of different alterations in different types of cancers, which are highly interesting for the biology of these diseases. And it's now also time for pan-cancer alterations. It's the NTRK alteration, it's the mutational burden, it's of course very classical, the morphology, and the microsatellite instability. So these can be found in many of these different cancer types. And if we just jump to the molecular drivers in the non-small cell lung cancer field, you can see there's a huge number of different drivers in this disease, and the majority is still unknown. And if you have a very careful look into the categories here, you see a very small frequency of these driver mutations in this disease. And it's more to be detected in the future. And if you translate this into histology on the right-hand side, you can also see that the non-small cell lung cancer is not only adenocarcinoma of the lung, but it's also squamous cell carcinoma and some other types. So it's broader than usually thought in the discussions. And the frequency, once again, now is in the perspective of different types of cancers, we can easily identify once again the non-small cell lung cancer as the most prominent category of cancers, but there is an emerging development at the moment that also other types of cancers are becoming more and more interesting in terms of molecular alteration and molecular subtyping of these diseases. What do we need to cover those broad molecular range of markers? Then we are very quick in the field of next generation sequencing, because Sanger sequencing, although this is a very nice technology, is not capable enough to detect multiple different alterations in one assay. And here, for example, the Oncomine Precision Assay is a tool which covers most or almost all of the relevant alterations in different types of cancers. So 50 genes have been compiled in this assay, covering almost 3,000 unique alterations, consisting of mutations, copy number variations, and fusion events. And most logical, the non-small cell lung cancer is very well represented here in the Oncomine Precision Assay. And here you can see that this assay also respects genes which are already labeled or in guidelines or even in the use of clinical trials. So you are able with this precision assay to answer a lot of relevant questions. A very emotionally discussed field in oncology in the last years, at least 2 and 3 last years, is the detection of fusion events causing fusion transcripts, which are nice targets for sequencing. However, it's sometimes a bit tricky if you have translocations and fusion transcripts where you know the driver gene but not the fusion partner. That always raises some technical concerns, how to cover this need to detect a fusion event on the RNA level without knowing the fusion partner. And this was done in the Oncomine Precision Assay in different ways. So it's quite easy for the known fusions where you know the driver and the partner gene. Then you can generate a system for amplification of this region, which is highly sensitive and very reliable. This is also the case if the sequence in between the driver and the partner is not known, you can also identify quite easily a system, an amplification system, which is able to have a sensitive amplification of this region, which is then very helpful for sensitive detection. It's more difficult and more tricky if the partner is unknown. Then you have only the driver situation, and then you need to have some bioinformatics tools for the interpretation of the sequencing data. These are RNA-derived data, in order to identify an imbalance situation. And there was much, let's say, advance in the last 1 or 2 years, in order to optimize these type of imbalance exon tiling system. And the Oncomine Precision Assay is a very well advanced system in this respect. But even the best gene panel for mutation detection or amplification or fusions, what we need in addition is a technology able to produce NGS data in a high throughput system in a very rapid time frame and very reliable. The preparation of the libraries used for next generation sequencing is to some extent quite cumbersome and lab intensive, and of course there is a high need based on a growing number of samples to do it very rapidly and with the support of automated system. And here we have a nice new development, which is the Ion Torrent Genexus System. It's fully automated. You can run it in 1 day and without much user intervention. So there's only a few hands-on time needed in order to run the entire system. So you start with the DNA or RNA, and the rest will be done by the machine, including sample purification, library prep, of course the sequencing, and the reporting of the data. So this system, this Genexus system, provides the user with a high degree of flexibility. So we can run different assays in parallel. We have multiplexing capability up to 32 libraries in a single run. We have a very nice feature, which is the use of the reagents for up to 2 weeks in the instrument, which is, I think, technicians will like it. We have a very nice colorful strips, so avoiding errors and mistakes in placing the reagents into the machine. And we have a quite small footprint for such an comprehensive machine in the lab, so we can really be quite restricted with the storage space we need. And in addition, which will come very soon, is a nucleic acid purification and quantification system. So even that will be added to the Genexus system to complete the entire workflow in a very, very elegant way. Of course, we are very interested, not only we in Berlin, but many sites across the globe, how this system is really working, because it sounds very fantastic to have a system which allows you to put in the DNA and then go away and let the machine work. So there was a decision for a multi-center study for evaluation of the Genexus system and the Oncomine Precision Assay. And this was done in Portugal, in Porto, in Switzerland, in Basel, at the Charité in Berlin, in Germany, and in Naples, in Italy. So we 4 centers joined forces in order to to test the Genexus system in combination with the Oncomine Precision Assay. We selected from each customer site own samples, which have been pre-characterized by other technologies or assays. So we cannot really compare across the sites the results, but we have, of course, the pre-characterization of these samples, and we have done FFPE samples, we have analyzed liquid biopsies, so the variant types, the mutations, the copy numbers, and the fusions. So quite broad approach for testing the Genexus system and the Oncomine Precision Assay. Let's start with the mutation analysis. As you can see here, we have included a number of different cancer types, altogether 45 samples, including some reference material, which you can see under the term unknown. I think what is also very important in the daily life is the knowledge regarding the tumor cellularity of each of the samples, because low tumor cellularity might prevent detection of important alterations. So that is always a challenge if it comes to very low tumor cellularity. That is the overview. That was our starting point. And then first of all, we checked for the representation of well-known alterations. You see here different types of EGFR mutations, PIK3CA mutation, KRAS mutations, and V2 mutations. And you can see there's a coverage, at least in this set of samples, which is quite convincing. And what is also a nice comparison is the allelic frequency. I mentioned in the previous slides that we also included samples with a low tumor cellularity, and this is also reflected here. You can see different alterations which are covered by this assay, and a really broad range of allelic frequencies. That is not only the tumor cell content, as you surely know, we have in many of these cancer types also tumor heterogeneity, which causes then low allelic frequencies. And of course, one of the most important parts of the study is the comparison with the already existing data. These have been done with the focus assay or Oncomine focus assay, the solid tumor Oncomine assay, AmpliSec colon and lung version 2 panel, and from a different vendor, the TrueSight 170. And on the right-hand side, you see a very nice correlation of the pre-existing data with those generated with the OPA and the Genexus system. And this comparison is summarized also in the small table. You see 100% specificity, and a concordance which is 100% or a little bit below 100%, which is very usual if it comes to the comparison of different technologies. Next, only a few numbers of cases with copy number alterations, because we restricted our comparison to already known alterations, and you can see here that this assay is also able to detect copy number variations, which is usually quite easy if you have a high copy number amplification, but it's more tricky, especially in combination with low tumor cell content, if there is a low copy number amplification. But this is done very nicely here with the OPA assay. You can see the comparison here with these type of amplifications. Of very high interest is the detection of fusion gene events based on the RNA level. You can once again see here that the tumor samples used for this assay is altogether 29 samples. Once again, the lung cancer is the most prominent one. again, important additional information is the distribution of the tumor cellularity, and this goes down to a range between 10 and 30%. The detection of fusion variants is represented here. You can see here a number of different fusion events, including the NTREX and the ALK, and some others with the known fusion partners. In addition, you can see here the MET exon 14 skipping event, also detected on the RNA level. Once again, the summary of these fusion events, you can see we included cases which were characterized by a variety of different assays, including the RGDX assay, the TrueSight 170, and of course the very traditional method, the FISH assay. And again, the specificity is extremely high, 100%, but also the concordance to already existing data is of really, really impressive high value. For the unknown fusion partners, I already mentioned at the beginning of this presentation, there was the development of a new bioinformatics algorithm in order to identify those unbalanced situation on the RNA. Here you can see some very nice examples how the detection of fusion genes works. In the upper level, you can see the targeted isoform detection, and this was done for 3 different samples, and you should realize that especially the NTREC1 and the red samples have a very low tumor content, so you can see only a limited number of reads, whereas the NTREC3 fusion was in a tumor sample with a very high tumor load. So that is the usual way it goes, but in the lower level you can also see the imbalance detection, which is nicely demonstrating that there is sufficient information available to identify not only the imbalance but also the location of the break in the gene, identifying the remaining kinase functionality, which is important for as a target for further analysis. So this approach allows you also in the case of unknown fusion partners to do a reliable detection of the fusion gene situation. And finally, also becoming more and more important are the liquid biopsies. You can see here from the numbers that we included 12 samples. Most of them have been derived from non-small cell lung cancer samples. Interestingly, in also our experience, it is not always possible to extract the recommended 20 nanograms of circulating tumor DNA. In this case, in our case collection of 12 samples, in some cases we were very low in the concentration and the amount of circulating tumor DNA. But let's see what comes out. You can see here that the distribution of these alterations in the plasma samples and the liquid biopsies, most of them not unexpected, are EGFR mutations, including the T7090M mutation. Of course, you will find KRAS and P53 mutation and some others. And as you can see, even in those cases with the very low amount of circulating tumor DNA, the detection was really successful. And finally, you can see here again the concordance with the pre-existing data, specificity 100% for all the alterations, and almost 100% concordancy for also for all the alterations. And this is also illustrated in the graph on the right-hand side, where you see a very high correlation with the pre-existing data. So, if we come to an overall summary and conclusion of the data, you can see here that the system, the Genexus, in combination with the Oncomine Precision Assay, is a very specific system. And also compared with a variety of different technology, we were able, with a high concordance and a high sensitivity, different types of alterations in formalin-fixed tissue samples, but also in liquid biopsies in the circulating DNA present in the plasma. So that is a very impressive result, and this was not from one single site. These are the data from 4 different sites across Europe. And I think this highlights to a very high degree the reliability of that approach. Let me conclude at the end. I think this increase of alterations is really demanding, and it's becoming progressively demanding in the future. So we have the challenge to cover these demands of the entire system. We need to be flexible in order to cover the diagnostic needs. We need to minimize the workload because technicians and scientists are quite expensive, and we have to save their working hours to do the data interpretation and not only the lab work. So automation is very welcome. And I think that the Genexus has a really great potential as a future solution to combine all steps of diagnostic next generation sequencing. And what we have seen in this study across different sites, the data are very robust and reliable, which provides an excellent basis also for the introduction in many more labs. Finally, I want to acknowledge the very excellent cooperation with the colleagues at the different sites. Also very nice communication and cooperation with the colleagues from Thermo Fisher Scientific, and of course from my team, that is Burkhard and Carsten, who really supported this work to a very, very broad extent. And thank you very much for your attention.
Thank you so much, Dr. Hummel, for that fantastic talk. We do have a few questions coming into the live Q&A portion of this presentation. Let me start with the first question. How do you address the recovery of good reads from FFPE samples, considering that these samples pass through a series of dehydration steps that might have significant effect to the DNA?
Well, that is a good question, but not a new question, because sequencing and next generation sequencing, which is applicable to DNA and RNA derived from formalin-fixed material, is already in use for a couple of years. What we see, if the tissue material is fixed under well-defined and good conditions, then we do not have a real problem using those kind of extracted DNA and RNA for a very, very good sequencing. Overfixation for a long time, or fixation in unbuffered formalin, or fixation at high temperatures is a very bad idea, causing DNA and RNA elements which are not really good for sequencing, irrespective of next-generation sequencing or Sanger sequencing. So I do not see a real problem using DNA and RNA extracted from good prepared formalin-fixed material.
Thank you for that. We have another question. Would you recommend the cell-free DNA samples as an alternative to tissue biopsy samples to avoid issues or concerns on tumor heterogeneity?
Good. Once again, a very good question. So the liquid biopsies are always thought to represent a kind of average of the tumor heterogeneity. However, that is not completely sure. So what we see usually is a very low amount of cDNA copies, cfDNA copies in the plasma, and it's not clear whether this is a full representation of the tumor heterogeneity. In addition, in some of the tumors where we know from imaging that tumor is in the body, there is no circulating tumor DNA detectable. So it's quite unclear so far whether this kind of average approach in the liquid biopsies is really reliable. So the idea is good, but I think we do not have enough knowledge currently to answer this question with the required reliability. I can add, if you want, so alternative to tissue biopsies, I think this is an addition, not a replacement of the tissue biospecimens. So yes, I like it very much, but as a kind of addition and not as a replacement.
Yes, absolutely. There's another question here. What is your take on panel-based NGS approach versus single gene testing, such as IHC of FISH?
Let's compare the immunohistochemistry with sequencing, with DNA-based sequencing. So we are talking about different types of molecules. So once for the immunohistochemistry, it's protein-based, using antibodies for the detection. Sequencing in many cases is DNA-based. So we have a different level of molecules. The FISH approach, again, is DNA-based, and we do not see in the FISH the consequence on the RNA and protein level. So we should always keep in mind that we are talking about different types of molecules with different function. For practical reasons, DNA is quite good to handle. It's a very good type of molecule for FISH, for DNA sequencing, and so it's often used in the lab. Immunohistochemistry on another side is antibody dependent, so you need very good antibodies to do a proper detection of your molecule of choice. If you use, for example, the BRAF antibody, yes, you can identify one certain type of BRAF mutation, but the sensitivity and reliability is not 100%. It's a much better approach to use the DNA because you have more information. FISH for the translocations, yes, that is very time consuming. It's a very nice technology, but once again, you do not see the consequence on the RNA and protein level. You see only the chromosomal breaks, which might be sufficient in a huge number of questions, but not always. What is the current trend? If you need to detect multiple different translocations because you do not know at the beginning of your investigation what might be the targeted fusion, then of course next generation sequencing based on RNA is a very, very good idea because you can identify a huge number of potential fusion genes in one assay. Regarding single or multiple gene detection, I'm very much in favor for the multi-gene detection because what we can see, in addition, not only the EGFR or the companion diagnostics approved molecules, we can also identify additional alterations, qualifying the patients for clinical trials. And I think this is a really, really major added value because that drives development to a very large extent.
Thank you so much for that insight, Dr. Hummel, and thank you once again for coming on board and presenting at our Genetic Solutions Tour. In the interest of time, I think we will end this session here, but we will now take a 15-minute break before the start of the next session. So grab a coffee, walk around the event, and check out the various application tracks that may be of interest to you. The clinical oncology session will continue after the break, but during this break, please visit our booth and learn about some very exciting promotions that are available to the audience and interact with our Thermo Fisher representatives at our booth who are right there to answer any of your questions. Please do not forget to check the leaderboard on the top right side of the screen to see what activities are available, including a scavenger hunt. Look out for our special icons in the environment to win prizes. There will be a pop-up survey appearing in a minute, so please do help us by completing this survey. Thank you again, and see you back in the auditorium in 15 minutes to continue our clinical oncology session.
Okay, bye-bye.
Thank you, Dr. Hummel.